Imagine standing on a cold mountain peak, maybe looking down into a vibrant deciduous forest, or perhaps you're floating above a coral reef. What's the first thing that really strikes you? Isn't it just the astonishing, almost overwhelming variety of life everywhere?
Absolutely.
From, a galloping horse, so powerful, down to the tiny, invisible molecular traffic inside a single cell. It makes you wonder, doesn't it? What actually is life?
And that sense of wonder, that huge question, is exactly where our scientific journey has to start. We move from just being amazed by life's diversity to asking, well, scientifically, what are the specific things that make something living different from non-living?
Right, the fundamental question.
Exactly. It's the bedrock, really, of all biology.
Welcome, everyone, to the Deep Dive. I'm your host, and joining me is our expert, ready to help us unpack the living world.
Hello.
Today, we're diving deep into the living world. Now, this is a foundational chapter, absolutely essential for your Class 11 and 12 boards, and crucially for cracking NEET and other medical entrance exams.
Very important.
Our mission today, to systematically look at life's diversity, figure out how we name it, and then how we classify it all. Think of this as your focused, step-by-step tuition session. We want every concept crystal clear for your exams.
Precisely. And our journey really begins by looking back. How did humans first try to understand life? Early humans, well, they were keen observers. They could instinctively tell the difference between inanimate stuff like wind, or the sea, fire, and living things like plants and animals around them.
Sure, this seems obvious.
It does now, but back then, this often led them to assign spiritual meaning, sometimes even deifying nature, living or not. It came from a place of awe, maybe sometimes fear. This emotional connection was, pretty constant.
So a different kind of understanding.
Very different. The move towards a systematic scientific description came much, much later. For a long time, societies had what we call an anthropocentric view of biology.
Anthropocentric, meaning?
Meaning centered on humans. Biology was seen mostly through the lens of human needs, food, medicine, shelter. How did other organisms help us? Okay, a practical viewpoint.
Understandable, but it limited progress. Real scientific advancement needed a shift away from this man-centric view towards something objective, something universal.
And what drove that shift?
Curiosity, fundamentally. A deeper need to understand nature for its own sake. This shift made it necessary to develop systems for identification, naming, nomenclature, and classification.
Right, bringing order to it all.
Exactly. And the most profound outcome, the real game changer from all this systematic study, was realizing that all living things share fundamental similarities.
Not just things alive now.
Not just now, but across all of time. The idea that every organism today is related somehow to every other organism today and to every life form that has ever existed. That was revolutionary.
Wow, that's a huge concept.
It really is. And understanding that interconnectedness sparked cultural movements, and it still drives global efforts for biodiversity conservation today. It highlights our shared heritage.
So, if I'm getting this right, that shift from a man-centric kind of utility-based view to a systematic, objective approach, that was absolutely fundamental.
Yes, completely fundamental.
What was the biggest hurdle that this systematic approach actually helped us overcome in terms of, advancing biological knowledge?
That's a great question. It really gets to the heart of it. The biggest challenge it overcame was, essentially, the lack of a universal way to understand and talk about life.
Communication.
Exactly. When knowledge was just based on human needs, it was fragmented, very localized, often inconsistent, no shared language, no standard methods. How could you build on discoveries or compare findings?
You couldn't, really.
Not effectively. Moving to a systematic approach allowed us to develop universal principles. Consistent identification, naming, grouping, no matter who was studying or where. That's what allowed biology to become a truly global science.
Okay, I see.
Now, when we ask that big question, what is life? There are really two questions hidden inside. First, the technical one, what defines living versus non-living? That's our focus today for your exams.
The scientific nuts and bolts.
Precisely. The second is more philosophical, the purpose of life. Interesting, but not our focus here. We stick strictly to the technical, the observable, the measurable.
Got it. Technical distinctions.
And to really grasp modern biology, especially evolution and classification, we absolutely have to talk about a key figure, Ernst Mayr.
Ernst Mayr. Okay.
Born in Germany in 1904. He was an evolutionary biologist at Harvard University, often called the Darwin of the 20th century.
I praise.
And deservedly so. He's widely seen as one of the 100 greatest scientists ever. His career spanned nearly 80 years.
Yeah.
He passed away in 2004, aged 100.
An incredible lifespan and career.
Truly. He joined Harvard in 53, retired in 75, but kept working as professor emeritus. His research was incredibly broad: ornithology, that's birds, taxonomy, zoo geography, animal distribution, evolution, systematics, even the history and philosophy of biology.
Wow, he covered a lot of ground.
He really did, a holistic view. But perhaps his most pivotal contribution was making the origin of species diversity the central question in evolutionary biology. It wasn't always the main focus before him.
So she shifted the focus.
He sharpened it, elevated it, and crucially, he pioneered the definition of a biological species that we largely still use today. This concept fundamentally changed how we categorize life.
The biological species concept. We'll need to unpack that.
We will. His impact was immense. He won what's called the Triple Crown of Biology, the Balzone Prize in '83, the International Prize for Biology in '94, and the Crayford Prize in '99.
Okay, clearly a giant in the field.
Undeniably, a testament to his lasting influence on our understanding of life itself.
That's an impressive legacy, no doubt. It sounds like Mayer's contributions were truly transformative for how we view species. You mentioned his definition of a biological species. Could you elaborate a bit more on why that was so significant? What was the paradigm shift it brought about?
Right, that's a crucial point. Mayer's biological species concept defines a species as essentially a group of natural populations that actually or potentially interbreed and are reproductively isolated from other such groups.
Okay, so focused on interbreeding.
Exactly, reproductive isolation. This was revolutionary because, before Mayer, species were often defined mainly by how they looked, their morphology.
Based on appearance.
Yes, but think about it. You could have two groups that look almost identical, but they can't interbreed successfully, can't produce fertile offspring. Are they the same species?
According to Mayer, no.
Correct. And conversely, think of dog breeds. They can look incredibly different, a Great Dane and a Chihuahua, but they can interbreed.
So they are the same species, Canis familiaris.
Precisely. Mayer's emphasis on reproductive isolation gave a concrete biological criterion. It went beyond just looking alike. It was testable. If two groups can't produce fertile offspring together in nature, they are distinct species.
Regardless of appearance.
Regardless of superficial similarity, this provided a much more robust framework. It helped map evolutionary pathways, understand how diversity actually arises. It shifted biology from just describing things to understanding the functional evolutionary processes, a real paradigm shift, and still a cornerstone today.
That makes the significance much clearer. Thank you.
Now let's shift back to that sheer, almost overwhelming diversity we started with. It's truly wonderful. The living world, the range of living types is just amazing.
The different habitats, too.
Extraordinary habitats. Cold mountains, forests, oceans, freshwater lakes, deserts, even boiling hot springs. Life finds a way, and it's awe-inspiring.
It really is.
And beyond the habitats, just the beauty. The grace of a galloping horse, migrating birds navigating perfectly, the colors in a valley of flowers, the raw power of a shark. It evokes awe, doesn't it?
Absolutely, a deep appreciation.
And it goes deeper still. Ecological conflict, cooperation within populations, between populations, and right down to the molecular level, that incredibly complex traffic inside a cell. It all forces us to think, what is life?
It's mind-bogglingly complex.
It is. Now, if you just observe your immediate surroundings, right where you are, you'll see a variety of organisms. Potted plants, insects, pets maybe, birds outside.
Sure, the everyday things.
Plus, countless others too small to see, microorganisms everywhere. Now imagine expanding your view. Go to a dense forest.
The variety would explode.
Exactly. Far greater numbers, far more kinds of living things.
And each different kind. That's a species.
That's our basic unit, yes. Each different kind of plant, animal, or microorganism you see represents a species.
Okay.
And when we count all these known species globally, the number scientists have actually described is somewhere between 1.7 to 1.8 million.
1.7 to 1.8 million.
A million, yes. This number refers to biodiversity, the number and types of organisms on Earth. But, and this is crucial, remember, this number isn't fixed.
It's still changing.
Absolutely. As we explore new places, use new technology, new organisms are constantly being identified and added to the list.
That figure, expert, 1.7 to 1.8 million. It's vast. But you say it's not fixed. Are biologists still actively finding new species, maybe even in places we thought were well known?
That's an excellent question, very perceptive. And yes, absolutely. The number is not constant, it's growing all the time. Biologists are indeed discovering new organisms constantly.
How? Where?
Well, several ways. Firstly, exploring places we couldn't easily reach before deep-sea vents, remote rainforest canopies, even microscopic worlds in soil, or inside us, our microbiome.
So new frontiers.
Definitely. Secondly, using new tools. Advances in molecular techniques like DNA sequencing are huge. They let us identify what we call cryptic species.
Cryptic species.
Organisms that look identical to known species, morphologically, but are genetically distinct. They can't interbreed. So they're separate species we just couldn't tell by looking.
Wow. Hidden diversity.
Exactly. So that 1.7 to 1.8 million, that's just what we've described. Estimates of the true total number of species.
Well, they range much higher, maybe tens of millions.
Tens of millions. We barely scratched the surface then. In many ways, yes. We're far from discovering everything. Biodiversity research is very much alive and ongoing.
Incredible. So how do we manage this huge, growing diversity? How do we talk about it?
That brings us to nomenclature and identification. Crucial processes. As we said, millions of organisms. We know them by local names, right? In our own area.
Sure. Like neem tree or sparrow.
Exactly. But those local names change from place to place, even within the same country, let alone globally. Imagine the confusion. If you say neem here, people understand. But talk to a biologist in Brazil.
They wouldn't know what you mean.
Precisely. So there's an absolute need to standardize the naming. We need a system so that a particular organism is known by the same name worldwide.
A universal name.
Yes. And that process of assigning these universal names is nomenclature.
Okay, nomenclature naming.
Correct. But you can only name something meaningfully if you know what it is. You have to describe it correctly, know its unique features, distinguish it from others. That preceding step, figuring out exactly what organism you're dealing with, is identification.
So identify first, then name.
You've got it. Identification leads to nomenclature.
And how is this standardized globally?
Scientists have established internationally agreed-upon procedures. These allow a unique scientific name for each organism, understood everywhere. For plants, the rules are in the International Code for Botanical Nomenclature, the ICBN.
ICBN for plants.
And for animals, there's a parallel code.
Yeah.
The International Code of Zoological Nomenclature, or ICZN.
ICZN for animals, got it.
The huge advantage of these scientific names, governed by these codes, each organism has only one unique name, no ambiguity.
One organism, one name.
Globally. And the system ensures that if you describe an organism properly, anyone, anywhere should arrive at the same name. It prevents confusion, duplication. A name once given can't be used for another organism. Uniqueness is key.
That makes perfect sense for global science. To make it even more concrete, you mentioned local names causing confusion. If I were to say AM in India for a mango, how would a biologist in, say, Europe know what I mean without this standardized system? Walk us through that practical problem.
That's a perfect example. AM. Instantly recognizable here in India, right? Millions know it.
Yes, very common.
But talk to a biologist in Europe or North America or anywhere outside the regions where AM is used.
Yeah.
They'd likely have no idea. They might guess it's a fruit, maybe, but which one? Total ambiguity.
So communication breaks down.
Completely. And that's where the brilliance of the standardized system comes in. The scientific name for mango is Mangifera indica.
Mangifera indica.
That name, Mangifera indica, following the rules of binomial nomenclature, which we'll detail next, is recognized unequivocally globally. A biologist in Europe sees Mangifera indica, they know exactly what fruit you mean. No guesswork.
It cuts through the language barrier.
Precisely. AM is local, cultural. Mangifera indica is the universal scientific identifier. It allows scientists from any background, any language, to talk about the exact same organism. It's like a common language, a lingua franca for biology.
A global language for life itself. That's powerful.
It really is. So building on that, biologists use these universally accepted principles. And the system is elegant. Each scientific name has just two parts.
Two parts?
Yes. The generic name and the specific epithet. This two-part naming system is called binomial nomenclature.
Binomial. That might mean two.
Exactly. Two names.
Yeah.
This system was introduced by the famous Swedish naturalist Carolus Linnaeus. And because it's so clear, simple, convenient, it's used by biologists everywhere.
Linnaeus. Okay.
Let's use mango again. Mangifera indica, the first word. Mangifera, that's the genus, a broader group.
Genus, all right.
The second word, indica, that's the specific epithet. It denotes the particular species within that genus.
Genus and species. Mangifera indica.
The unique combination makes the full name. Now, for your exams, NEET, boards, Knowing the rules of nomenclature is absolutely critical. Let's go through them step by step. Very important.
Okay, let's focus here. The rules.
Rule 1. Biological names are generally in Latin, and they're written in italics in print. Even if the word comes from another language, it's Latinized. Mangifera indica italics. Latin gives it stability, a universal foundation.
Okay, Latin italics, rule 1.
Rule 2. The first word is the genus. The second word is the specific epithet. Genus first, species second. Always that order.
Genus, then species. Got it. Rule 2.
Rule 3. When you handwrite a biological name, both words must be separately underlined. Pay attention to this for exams. If it's printed, it's in italics. Underlining or italics indicates the Latin origin. Crucial detail.
Handwritten, underlined separately. Printed, italics. Okay.
Rule 3. Rule 4. The first word, the genus, always starts with a capital letter, so M in Mangifera. But the specific epithet, the second word, always starts with a small letter. The idea in indica is lowercase. Capital genus, small species, distinct rule.
Capital genus, small species, rule 4, very clear.
And one more point. Often the name of the author who first described the species comes after the specific epithet, usually abbreviated, like Mangifera indica Linn.
Linn for Linnaeus.
Exactly. It indicates Linnaeus first described it, gives credit where it's due. Academic standard.
That's incredibly clear, breaking down each rule like that. But let me ask about a common mistake students make. Based on these rules, if a student writes Mangifera indica in an exam with a capital I for indica, would that be marked incorrect? And why, based on the rules?
Yes, absolutely. Writing Mangifera indica with a capital I would definitely be considered incorrect. It's a very common error, unfortunately.
For a reason.
It directly violates Rule 4, which we just covered. Rule 4 explicitly states, the specific epithet starts with a small letter. Only the genus name, Mangifera, gets the capital letter. The specific epithet, indica, must start with a lowercase letter.
So it's not optional, it's a strict rule.
It's a strict, internationally agreed upon convention. Getting it right shows you understand the fundamental principles of biological nomenclature. That's what examiners look for: precision and adherence to these universal standards.
Okay, crystal clear. Capital I in indica is wrong. Noted.
Good. Now moving on from naming, how do we organize this vast diversity? We can't study 1.8 million organisms one by one.
Right, impossible.
So we need a system to make sense of it all. That process is classification. Basically, classification is just grouping anything into convenient categories based on shared, easily observable characteristics.
Grouping things based on what they have in common.
Exactly. Think about everyday life. We instinctively group things: plants, animals, dogs, cats, insects. When we use those words, certain characteristics immediately come to mind, right?
Yes. You picture a dog, not a cat.
Precisely. Or think of Alsatians, German Shepherds. You know exactly which kind of dog. Or mammals. You think of animals with fur or hair, maybe external ears.
Right. Shared features define the group.
Or for plants, wheat. You picture a wheat plant, not rice. So all these terms, dogs, cats, mammals, wheat, rice, plants, animals, they're convenient categories for study.
And the scientific term for these categories?
The scientific term is taxa, singular, taxon. And it's crucial to realize taxa exist at different levels. Plants is a taxon, a very broad one. Wheat is also a taxon, but much more specific, within plants.
Like nested boxes.
A good analogy. Animals, mammals, dogs, all taxa, but at different levels in a hierarchy. Dog is a type of mammal, mammal is a type of animal, each level defined by shared characteristics.
Okay, taxa are the categories at different levels.
And the whole process of classifying living organisms into these different taxa based on their characteristics is known as taxonomy.
Taxonomy, the science of classification.
Yes, and modern taxonomy isn't superficial. It's based on deep biological details. External structure, internal structure, anatomy, cell structure, cytology, how organisms develop, embryology, and very importantly, their ecological information, how they interact with their environment and other organisms.
So it's comprehensive.
Very comprehensive. It uses all these aspects. So to sum up taxonomy, it involves 4 fundamental processes, all linked. First, characterization, describing the organism's traits. Second, identification, determining what it is, matching it to known groups or recognizing it as new.
Identification.
Third, classification, placing it into the correct categories based on those characters.
Classification.
And fourth, nomenclature, giving it the correct scientific name according to the rules.
Nomenclature. Characterization, identification, classification, nomenclature. The core of taxonomy.
Precisely. Those 4 steps are the foundation.
You mentioned earlier that early classifications were often based on uses for humans. how does modern taxonomy, with its focus on all these detailed biological characteristics, cell structure, development, ecology, how does it fundamentally differ from those initial utility-driven ways of grouping things? What's the core shift?
That's a critical distinction. Early classifications were, as we said, largely pragmatic, anthropocentric. Humans were interested in organisms mainly for their own needs: food, clothing, shelter.
Practical needs.
Exactly. So plants might be grouped as edible, medicinal, poisonous, animals as huntable, domestic, dangerous. The criteria were all about their use or impact on humans. Modern taxonomy. Radically different approach. While utility might still be noted somewhere, the fundamental basis for classification is the inherent biological characteristics of the organisms themselves.
Regardless of human use.
Primarily, yes. We look at external form, internal anatomy, cells, embryos, genetics, DNA, RNA, ecological roles, everything. The goal is to understand the natural relationships between organisms based on shared ancestry and evolution.
So it reflects biology, not just human perspective.
Precisely. It aims for an objective, comprehensive system that reflects evolutionary history, not just human convenience. This allows for a system that's predictive, consistent, and scientifically rigorous. It reflects biological reality far more accurately.
That's a major shift in thinking.
A huge shift. Now, humans haven't just been interested in listing organisms and their diversity. For a long time, we've also been fascinated by the relationships among them.
How they're connected.
Exactly. And this branch of study, looking at diversity and relationships, is called systematics.
Systematics.
Okay. The word comes from Latin, systema, meaning systematic arrangement. Linnaeus actually used Systema Naturae as the title for his major work on classification.
Ah, linking back to Linnaeus.
Yes. Now, initially, systematics was mostly about classification, but its scope broadened. It now includes identification, nomenclature, classification, the core of taxonomy, but with a crucial added dimension.
Which is?
It takes into account evolutionary relationships between organisms.
Ah, the history. The family tree.
Precisely. Systematics doesn't just group things by similarity. It tries to understand why they're similar, based on shared ancestry and evolutionary history. It puts classification into a phylogenetic context.
So if taxonomy is about the what and how of classifying and naming, is it fair to say systematics adds the why, the evolutionary history and relationships? Is that the core difference?
Exactly. That captures it perfectly. Taxonomy provides the framework, characterization, identification, naming, grouping. Systematics uses that framework but asks the deeper questions. How are these groups related through evolution? What's their shared history? How did this diversity arise?
Okay.
So yes, systematics adds that vital dimension of evolutionary history and relationships to the classification. It connects the present diversity to its deep past.
Understood. Taxonomy is the structure, systematics adds the historical narrative.
Well put. Now, let's look closer at how this classification structure actually works. It's not just one step. It involves a hierarchy of steps.
A hierarchy, like ranks.
Exactly. Each step represents a rank or category. Since each category is part of the overall system, it's called a taxonomic category. And all the categories together, in their sequence, form the taxonomic hierarchy. Each individual category, which acts as a unit of classification, represents a rank and is commonly called a taxon, plural, taxa.
So taxon is the word for a specific category or rank.
Correct. Let's use an example. Insects. That's a recognizable group, right? They share common features like 3 pairs of jointed legs.
Yes.
So insects represents a distinct group, a biological entity. It can be classified. It's given a rank, a category. It's a taxon. Other groups, like birds or fish, are also taxa, categories at a certain rank. These aren't just random collections. They are distinct biological entities defined by shared characteristics reflecting evolution.
Got it.
Now, through decades, centuries really, of studying organisms, scientists developed a set of common, universally used categories. Listed in order, let's say, from broadest down to most specific. Kingdom, phylum, or division for plants, class, order, family, genus, and species.
Kingdom, phylum, division, class, order, family, genus, species. Okay, need to remember that order.
Absolutely crucial for exams. And remember, species is the lowest, most fundamental category, the basic unit for all organisms.
Species is the base level.
Yes. Now, how do we actually place an organism into these categories? The key requirement is knowing its characteristics thoroughly.
The details matter.
Immensely. Understanding the similarities and differences helps us identify relationships among individuals of the same species and between different kinds of organisms. That allows us to place them accurately in this hierarchy.
Excellent. This hierarchy is key. For everyone listening, especially if you're prepping for exams, try mentally listing those categories right now, maybe highest to lowest, before the expert dives into each one. Good way to check your recall.
That's a great suggestion. Active recall is very helpful. Okay, let's explore each category now, starting from the bottom, the most fundamental unit, and working our way up. 9.1 species, 1.2.1. So taxonomic studies consider the species as the fundamental unit. It's defined as a group of individual organisms with fundamental similarities.
Meaning they share core traits.
Yes, a shared gene pool, basic structure, function. Crucially, they can interbreed in nature and produce fertile offspring. That's key to the biological species concept.
Fertile offspring.
Right. And importantly, when you compare one species to another closely related species, you should be able to distinguish them based on distinct morphological differences.
Observable physical differences.
Exactly. Consistent physical traits that let us tell them apart, even if they look quite similar overall.
Okay. Can we have some examples?
Definitely.
Yeah.
Let's take Mangifera indica, the mango. Here, indica is the specific epithet defining this species, or Solanum tuberosum, the potato. Tuberosum is the specific epithet, and Panthera leo, the lion. Leo is the specific epithet.
So indica, tuberosum, leo, those are the species names. Well, epithets.
The specific epithets, yes, they represent the species level. The first words, Mangifera, Solanum, Panthera, those are the genera. The genus, remember, is the next level up.
Genus is broader than species.
Correct. And a single genus can contain multiple species. Different specific epithets representing different organisms, but they share enough similarities to be in the same genus.
Like different models from the same car brand?
Sort of.
Yeah.
For instance, the genus Panthera includes Panthera leo, lion, but also Panthera tigris, tiger. Different species, clearly, but very closely related morphologically and genetically.
Okay. Other example.
Genus Solanum. It has Solanum tuberosum, potato, but also Solanum nigrum, black nightshade, and Solanum melongena, brinjal lake plant. Distinct species, but grouped in Solanum because of shared features, especially in their flowers.
Right.
And for us, Homo sapiens. Sapiens is our species. Homo is our genus. It shows how distinct species fit within broader generic groups.
So, expert, the key differentiator for a species, even if they share broad similarities within a genus, it really boils down to those distinct morphological differences plus the reproductive isolation. Is that the main takeaway for students?
Precisely. While species in the same genus share many common features more than species in different genera, it's those distinct morphological differences, combined with reproductive isolation, not being able to interbreed and produce fertile offspring naturally, that truly define and separate one species from another.
Even if the differences seem small sometimes.
Even that. These differences are consistent, genetically based, and ecologically significant. They maintain the integrity of the species line. So yes, focusing on distinct morphological differences and reproductive isolation as key species identifiers is the crucial takeaway. 9.2 genus, 1.2.2. Moving up from species, we get to the genus. A genus is essentially a group of related species.
Related species grouped together.
Exactly. And the key idea is that the species within one genus share more characters in common compared to species belonging to other different genera. You can think of a genus as an aggregate, a collection of closely related species.
More similar to each other than to anything outside the group.
Precisely. Let's use our examples again. Potato, Solanum tuberosum, and brinjal, Solanum melongena. We know they're different species,
Right.
but both belong to the genus Solanum. This shows they share a significant number of fundamental traits, especially plant structure and flower patterns, grouping them together.
Despite their species-level differences.
Yes. Similarly, think about the big cats. Lion, Panthera leo, leopard, Panthera pardus, tiger, Panthera tigris, all distinct species,
Utterly.
but they share many common features: carnivorous, predatory, broad morphology, so they're all placed in the genus Panthera. This genus, Panthera, is then distinct from another related genus, Felis, which includes domestic cats and smaller wild cats.
So Panthera and Felis are different genera, but related.
They are related, yes. They belong to the same family, which we'll get to next. But the differences between, say, a tiger and a house cat are significant enough to place them in separate genera, Panthera and Felis, reflecting distinct sets of shared characteristics at the genus level. 9.3 family, 1.2.3. Okay, next level up. The family. A family is a group of related genera.
So related genera make up a family.
Correct. And remember the pattern. As we go higher up the hierarchy, the number of shared similarities decreases. Genera within family are related, yes, but they share fewer specific common characteristics compared to the species within a single genus. The connections are broader.
Similarities become less numerous, more general.
Exactly. Now, for plants, families are characterized using both vegetative features like roots, stems, leaves, and, very importantly, reproductive features, especially the structure of their flowers.
Flowers are important for plant families.
Very important. Floral characteristics are often more stable and reliable indicators for classification at this level.
Okay, examples.
For plants, 3 different genera: Solana potato, brinjal, petunia, and datura. Despite being distinct genera, they are all placed together in the family Solanaceae.
The nightshade family.
That's the one. They share fundamental characteristics, especially in their flower structure, like 5-part flowers, that link them. For animals, the genus Panthera, lion, tiger, leopard, is grouped with the genus Felis, cats, into the family Felidae.
Felidae, the cat family.
Right, all cat-like animals sharing traits like retractable claws, sharp senses, predatory behavior. Now, compare a cat, Felidae, with a dog. Some similarities as mammals, sure, but also big differences in skull, teeth, behavior.
Very different.
Different enough to be in separate families. Cats are in Felidae, dogs are in Canidae. This shows how families group related genera based on broader yet still significant shared traits reflecting deeper evolutionary ties.
For plant families, you mentioned both vegetative and reproductive features. In practice, expert, do taxonomists usually give more weight to one over the other, or is it generally a balanced consideration?
That's an insightful question, and yes, there's often a weighting. While both types of features are considered, reproductive features, particularly floral characteristics, are generally given more weight when classifying plant families.
Why is that?
Primarily because floral structures tend to be much more stable and less affected by environmental changes compared to vegetative parts. Think about it. Leaf size or plant height can vary a lot depending on soil, water, light.
True.
But the fundamental structure of the flower, number of petals, sepals, stamens, the arrangement of the ovary, these are typically highly conserved, very consistent within a family. They're considered more reliable indicators of true evolutionary relationships.
So more genetically stable, less plastic.
Exactly. So while all evidence is weighed, floral characters often provide the more definitive evidence for placing a plant genus into a particular family. 9.4, order 1.2.4. All right, continuing our ascent. The order. Now, you've seen that species, genus, and family classifications rely heavily on specific, similar characters. When we get to order and higher categories, we generally base identification on aggregates of characters.
Aggregates, meaning like a collection of broader traits.
Precisely. A collection or combination of broader characteristics. The number of shared specific features becomes much smaller, so we look at overall patterns. And order, being higher than family, is an assemblage of families.
Grouping related families together.
Yes. Families within an order exhibit a few similar characters, but these similarities are definitely fewer compared to the similarities you'd find among genera within one family. The relationship is becoming more distant. Okay, for plants. Families like Convolvulaceae, morning glories, and Solanaceae, the nightshade family we discussed, are both placed in the order Polemonialis. This grouping is largely based on shared floral characters that indicate a broader relationship.
Polemonialis, okay. And animals.
A classic example is the order Carnivora. This order includes families we've mentioned, Felidae, cats, and Canidae dogs.
Both are carnivores, makes sense.
Right. They share aggregated characters related to their meat-eating lifestyle, sharp teeth for tearing flesh, specific digestive systems, even though they belong to quite distinct families. 9.5 class, 1.2.5. Moving higher still, we reach the class. This category includes related orders.
Grouping related orders now.
Exactly. The common characteristics become even broader, more fundamental, linking organisms with a more distant but still significant evolutionary past.
Example.
A key example for animals is the class Mammalia. This class includes the order Primata, monkeys, apes, humans. It also includes the order Carnivores, tigers, cats, dogs.
So primates and carnivores are in the same class.
Yes, Mammalia. And it includes many other orders too. Rodentia, rodents, cetacea, whales, and so on. What unites them all in class Mammalia?
Milk, hair, warm-blooded.
Excellent. Presence of mammary glands, body hair, being warm-blooded, homeothermic, usually a 4-chambered heart. These are fundamental mammalian traits shared across all those diverse orders, pointing to a common ancestor. 9.6 phylum, 1.2.6 division for plants. Next up is the phylum. This groups together related classes.
Grouping classes now, like...
For animals, think about classes like fishes, amphibians, reptiles, birds, and mammals. All these diverse classes are put together in the next higher category, the phylum.
What unites such different groups? Fish and mammals seem worlds apart.
They seem different, but they share fundamental body plan features, usually present at least during embryonic development. For instance, all those classes are grouped into phylum Chordata,
Chordata, what's the key feature?
based mainly on having at some stage a notochord, a supportive rod, a dorsal hollow nerve cord, which becomes brain and spinal cord, and pharyngeal gill slits. These deep structural similarities indicate a shared evolutionary origin.
Notochord, dorsal nerve cord. Okay, fundamental structure.
Exactly. Now, important note for exams. For plants, the category equivalent to phylum is called division.
Ah, right. Phylum for animals, division for plants.
Correct. So plant classes with similar fundamental characters are grouped into a division. For example, all flowering plants belong to the division Angiospermae. The principle is the same grouping related classes, just the terminology differs.
So phylum for animals, division for plants, but both serve the same purpose. Grouping related classes based on fundamental similarities. Is that correct summary?
That is an absolutely perfect summary. Exactly right. Same hierarchical level, same function of grouping related classes based on broad fundamental features, just different traditional terms for the animal and plant kingdoms to maintain clarity. 9.7 kingdom, 1.2.7. And finally, we reach the top. The highest category in the taxonomic hierarchy is the kingdom.
The broadest category of all.
The broadest. In animal classification, all animals belonging to all the various Phylochordata, Arthropoda, Mollusca, etc., are assigned to the highest category, kingdom Animalia.
Kingdom Animalia, all animals.
Yes. Multicellular, heterotrophic, typically mobile, no cell walls. They're defining traits. And distinct from that is kingdom Plantae. This comprises all plants from various divisions, Angiospermae, Gymnospermae, ferns, mosses.
Kingdom Plantae, all plants.
Generally speaking, yes. These two, the animal and plant kingdoms, are the two broadest groupings of life recognized in many traditional systems, representing vast swaths of evolutionary history.
Okay, kingdom is the top level.
Now, let's consolidate this whole hierarchical idea. Imagine that ladder again. From species climbing up to kingdom, there's a crucial principle often tested. As you go higher up the hierarchy, from species towards kingdom, the number of common characteristics shared by the members goes on decreasing.
Fewer similarities at higher levels.
Exactly. Species members share the most. Kingdom members share only very broad fundamental traits. Conversely,
The lower the taxon, the more shared characteristics.
precisely, members of a species share tons of features, much more than members of the same genus and vastly more than members of the same kingdom. It's an inverse relationship. Higher category, fewer shared specifics. Lower categories, more shared specifics.
Okay, that makes sense. High level, general similarities. Low level, detailed similarities.
They got it. And this has an important implication. The higher the category, the greater the difficulty in figuring out the precise relationships to other taxa at that same high level.
Why is that?
Because the shared characteristics become so broad and general, it's harder to find unique defining features or pinpoint exact evolutionary branching points. Classifying the kingdom level is conceptually more complex than at the species level.
More challenging to draw fine lines.
Exactly. Which is why taxonomists have also developed subcategories like subfamily, superorder, to allow for more precise placements within this main hierarchy.
That inverse relationship higher category, fewer shared traits, can be tricky for students. Could you maybe re-emphasize it with a simple analogy? Perhaps comparing, say, a family of students in one class versus a kingdom of all students in the whole district.
That's an excellent analogy. Let's try that. Imagine your school system is the hierarchy. Think of a family, maybe your close study group, 5 friends in class 11 biology.
Okay, a small, specific group.
Right. What do they share? Same class 11, same subject biology, same school, probably live nearby, share specific academic goals, maybe even inside jokes.
Yeah.
Loads of common characteristics, right?
Yeah,
Very specific ones. Because it's a low taxon, a closely related group.
makes sense. Lots in common.
Now, compare that to the kingdom of all students in the entire school district. This includes everyone from class 1 to class 12, different schools, huge age range, diverse interests spread across the whole district.
A massive, diverse group.
Exactly.
Yeah.
What are the common characteristics all of them share? Well, they're all students. They all attend school. They all live in the district. Very broad, very general characteristics.
Not much specific detail shared by everyone.
Not at all. The specific things binding the study group family are completely diluted at the district kingdom level. So the analogy holds. Lower taxon, study group family, many specific shared traits. Higher taxon, district kingdom, few broad general shared traits. As you go up, shared specifics decrease, making relationships harder to pin down precisely at those higher levels. That conceptual difference is key.
That analogy really helps clarify it. Thank you.
Good. Now, to really nail this for your exam, let's apply these categories to specific organisms. We'll use the examples often found in textbooks, like in Table 1.1 man, housefly, mango, wheat. Knowing these classifications is critical for recall.
Absolutely, expert. Let's take these one by one. For our NEET aspirants, this table comes up often fill in the blanks, matching, finding errors. Could you systematically walk us through the full classification for each? Species to kingdom.
Certainly. Let's be precise. First, man, Homo sapiens, species, genus, homo, family, hominidity, order, primata, class, mammalia, phylum, chordata, kingdom.
Animalia.
Okay, man. Hominidae, primata, mammalia, chordata, animalia. Got it.
Next, housefly, Musca domestica. Species, Anica, Mesca, Musca. Family, Muscidae, order, Diptera, class, Insecta, phylum, Arthropoda, kingdom, Animalia.
Housefly, Mysidae, Diptera, Insecta, Arthropoda, Animalia.
Right.
Now, a plant, Mango 4, Magifera indica, species, Indica, genus, Angifera, family, Anacardiaceae, order, Sapodilus class, Dicotyledone, division, Angiospermae, remember, division for plants, king.
Mango, Anacardiaceae, Sapodilus, Dicotyledone, Angiospermae, plantae.
Okay, and finally, wheat, Triticum aestivum, species, genus, Triticum, family, Poaceae, grass family, order, Poiles, class, Monocotyledone, division, Angiospermae, kingdom, plantae.
Wheat, Poaceae, Poas, Monocotyledone, Angiospermae, plantae. Perfect.
Memorizing these examples really helps visualize how organisms fit the hierarchy, and it reinforces that point about commonalities decreasing as you go up the ranks. Crucial examples for understanding and for exam success.
Definitely worth committing those to memory.
So let's quickly summarize the key points from our deep dive into the living world. We started by appreciating the sheer, amazing diversity of life. Millions of species described, many still unknown. This diversity pushes us to define what living actually means.
The fundamental question.
To study this diversity systematically, biologists developed universal rules for identification, nomenclature, especially binomial nomenclature, and classification. The science dealing with all this is taxonomy.
Identification, nomenclature, classification, taxonomy.
We stressed binomial nomenclature Linnaeus, the two-part name, genus, species, and those 4 crucial rules, governed by international codes, ICBN, ICZN, for global consistency.
The rules are key.
Then we explore the taxonomic hierarchy, the ladder of classification from species up to kingdom. Species, genus, family, order, class, phylum, division, kingdom. And the vital principle. Common characteristics decrease as you go up the hierarchy. Lower taxes share more specifics.
That inverse relationship.
Exactly. And these systems aren't just academic. They're vital for agriculture, forestry, industry, conservation, for understanding and managing our planet's biodiversity.
It connects to everything.
It really does.
Yeah.
It's foundational knowledge.
That was an incredibly thorough and clear explanation, expert. Thank you. For all our listeners, NEET aspirants, Class 11 and 12 students, this chapter is non-negotiable foundational knowledge. It really sets you up for more advanced biology. To really master this for your exams, we strongly recommend you revise this topic again. Focus on the definitions, and especially the examples for each taxonomic category we covered.
Yes, the examples help solidify understanding.
And definitely practice past year questions. Look for questions on binomial nomenclature rules and those specific classifications from Table 1.1 man, housefly, mango, wheat. Those come up frequently. Your ability to recall and apply these will make a difference.
Absolutely. And remember, understanding the living world isn't just about memorizing facts for an exam, though that's important right now. It's also about starting to appreciate the incredible order, the intricate connections, the vast web of life on Earth.
A bigger perspective.
Exactly. Keep your curiosity alive. Don't hesitate to go back over things if you have doubts about this chapter. Keep learning, keep asking questions, and keep growing.